So today I'm going to be talking about learning without a brain. And before I jump into the meat of the topic of the lecture, I first want to acknowledge that all of the work I'll be presenting today is a collaboration with these wonderful folks, especially my PI, Wallace Marshall at UCSF, and my co-PI, Adam Frost, who's now at Altos Labs. This is also a collaboration with the Center for Cellular Construction and these wonderful summer course students that I had the pleasure of working with last summer. So at this point, I know I can't see all of you, but I'd like all of you, the comfort of your homes, to take a moment and close your eyes. Yes, close your eyes as I'm doing now. And I'd like you to notice if you feel anything on your skin. The itch of fabric, the texture of cloth, the softness of your shirt. You can open your eyes now. So you probably notice the feeling of your clothes on your skin when you put them on in the morning. But over the course of the day, you likely learn to ignore them because you have more important things to focus on, like these mini medical school talks. And what you've just experienced is a form of learning called habituation. So habituation is a form of learning in which a response decreases after repeated stimulation. And in your case, the repeated stimulation was the rubbing of fabric against your skin. But the thing is, you have a brain with 86 billion neurons to help you with this feat of learning. And I'm interested in how even a single cell, the cell that you can actually see in my Zoom background, is capable of the same type of learning despite not having a brain. So now let me introduce the charismatic stentor ceruleus. This is the same video playing in my Zoom background. And this is a single cell that is also capable of habituation. So a little bit of background about stentor ceruleus. So this is a unicellular organism that we can find in ponds, and I sometimes collect them locally, even from Golden Gate Park. It's a protist, and it's actually a giant cell. This trumpet-like morphology is 1 to 2 millimeters long, which is quite large for a cell. It's about the size of a period at the end of a sentence. And stentor got its name from a character in the Iliad, the epic. So in the Iliad, there is this herald trumpeter named Stentor. And because stentor is trumpet shaped, that's how it got its name. And the second part, ceruleus, comes from its cerulean blue-like color. So going a bit into the anatomy of stentor, so here we have a single stentor. What we saw before was a group of stentor aggregated together, as they sometimes do to help with their feeding behavior. So at the top, we have the membranellar band. And the membranellar band has a lot of cilia. These are hair-like appendages that help with the flow of food into the cell. We also have the nuclei, which are sort of like pearls on a string type of shape. And this is what controls, basically has all the instructions to rebuild the cell. And I'll get into this a little bit later, but stentor has the amazing ability to regenerate once you cut or injure it. And they have many ciliary rows, which are stripes along the axis of the stentor that help with its contraction. And I'll show the videos of that in a few slides. And they have a hold fast, which allows them to stick to the surface that they're on, the surface of a pond or the surface of vegetation that they find in the pond. When I mentioned stentor contraction in the previous slide, when stentor get a tap or any sort of mechanical stimulation, they contract from that elongated trumpet-like shape to a ball-like shape, sort of like a roly-poly, and this is an escape response from predators. But as I mentioned, the cool thing about stentor is that they're capable of learning. They're capable of habituation, just like you did with the clothing on your skin. So when stentor get repeatedly tapped, they actually learn to ignore it, and they remain in this elongated state, whereas the naive cells without any training continue to contract. So as you can see here, this is the normal extended trumpet-like shape. And when they get a tap, they all contract, looking at as this mass over here. So I just want to briefly mention what is the context of stentor in the wild, because even though I do a lot of work in the lab, I think that so much of biology is useful in the context of what's the natural history of the creature, what are its natural predators and prey. So contraction is an escape response from predators in the pond. So this is a sample that I actually got from Golden Gate Park. This is actually Lake Merced over here that I'm collecting the sample from, but these guys here are from Golden Gate Park. And you can see these Carididae creatures voraciously snacking on the stentor. If the stentor perhaps had contracted fast enough, it would have been able to escape. But contraction is actually adaptive and helps the stentor escape from these critters here, as well as a number of other predators, including me. I could be a predator too. Some people have sequenced the guts of humans and found traces of stentor. So next time you drink out of a pond, you might find some stentor inside of you. But the thing is, if a stentor is constantly contracting in response to harmless stimuli, such as ripples in the pond that are not actually predators, then the stentor would be wasting energy. So habituation is also adaptive. It helps the stentor preserve energy so that it's not contracting away from innocuous stimuli. So this is what a group of stentor look like when they're contracting. Each of these is a single cell, and they're receiving a tap, and you can see them contracting almost instantaneously. So I showed you where stentor are found in the wild, but to recapitulate a lot of this in the lab, we've built a habituation device, which is basically a device that can deliver repeated taps to stentor at a specified force and frequency. So it's automated, so I don't have to sit there and use my finger to tap the cells all the time. So this Petri dish here contains the cells in the video that you saw in the previous slide. So all of them fit in this tiny little dish. And then this is the metal strip, the armature that is repeatedly hitting the ruler atop which the Petri dish rests. And this is an Arduino controlled device, so everything is automatic. And the USB microscope is positioned over the dish so that we can get the videos like you saw in the previous slide. So here you can see the metal strip is hitting the ruler. It's a close-up, and this is the dish containing the stentor. So that's what it looks like. The mechanical tap is delivered to the entire dish at once, and all of the cells are responding. Here's the same group of cells I showed you before, but you can notice a difference probably, which is that now that they're receiving the tap, not all of them are contracting. In fact, most of them are not contracting. So this group of stentor has received training. They've been repeatedly hit by the metal armature on that habituation device for 60 minutes at a frequency of 1 tap per minute. So with all of that training, they've actually habituated to these weak force taps. And so they've learned to ignore it, and most of them are no longer contracting. And when we graph this is what it looks like. So at time 0, almost all the cells contract in response to the mechanical stimulus, the tap from the device. And by the end of the experiment, almost none of the cells are contracting. So they have learned to ignore the stimuli. It's really amazing that a single cell can do what we can do, but without a brain. And it begs so many different questions. And here are just a few of them that I'm going to touch on today if we have time. So first of all, what I showed you was a group of cells in a dish. So I'm showing you what learning looks like at the level of a population. But one question I had was, how does this population level behavior emerge from single cells? How is each individual cell in that Petri dish actually learning? And then when the cell goes from a naive state to a learned state, how does that happen? What are the factors or input stimuli that influence that transition? And then how does this learning even occur? Do they have a mini brain, a series of different molecules inside the cell that help with learning? And finally, how do other types of stimuli impact stentor behavior? So of course, the pond is a very messy place. There are all kinds of other stimuli from other animals, from ambient light. And I was wondering, how do all these other factors influence their learning? So first, the first question, how does this population level behavior emerge from single cells? So going back a couple of slides, I showed you this gradual decline in contraction probability. So on the x-axis, we have number of stimuli, that the number of taps that the cells are getting. And on the y-axis, we have the fraction of cells in the population contracting. So there are two main possibilities that emerge here. Either each cell is learning gradually, or each cell is undergoing a discrete switch from a naive state to a learned state, but the switch could occur at different time points for each cell, which would lead to this emergent population level decline. So the first order of business for me was to figure out which of these two possibilities was happening. So to answer this, basically, I analyzed the contraction behaviors of each individual cell. So in each cell of the dish in the videos I showed you before, I looked at every single time point. Is the cell contracting or is it not contracting? And here are just two representative examples, two cells in the video I showed you before. So on the x-axis, again, we have stimulus number, so the number of taps the cells are getting. And on the y-axis, we have probability of response. And each black square represents an instance in which the cell received a tap and contracted. And the white squares represent an instance where the cell received a tap and then did not contract. It remained in that extended state. So just visually looking at this, you can see there appears to be a discrete, a sudden switch from when the squares are mostly black to the squares being mostly white. And to confirm that this is actually a single switch, we implemented an algorithm at every single time point to test whether there is a significant difference in contraction probability before and after that time point. And we found that this is indeed the case. And this occurs at different time points for each different cell. So for example, for cell number 1 up here, that switch happens before tap number 10. Whereas for the second cell here, that switch occurs before tap number 20. So this is basically like a eureka moment for each single cell. Instead of gradual learning, there's a discrete switch when the cell switches from mostly contracting to mostly not contracting, or in other words, from naive to habituated. So to basically confirm this, we did a Fisher's exact test, again, to look at the various time points, is there a significant difference? And we found that there is a low p-value for the actual data compared to the scrambled data, showing that there is a significant step that's occurring from in a difference in behavior between the naive state and the learned state. And we also wanted to test, is there actually only a single step happening, or are there multiple steps happening? So we repeated this analysis for all of the time points before the calculated switch, and then all of the time points after the calculated switch. So basically pre-step and post-step here. And we found that there is no significant difference. So what this means is that there is a single switch, and it is significant between the naive state and the learned state, the eureka moment for the cell. So we can summarize all of the information I just said in this simple diagram. We can describe stentor habituation as a two-state model. In P1, we can call this the responsive state, when the cell is mostly contracting in response to getting the taps. And P2, we can call it the non-responsive state, when the cell is mostly not contracting when they're getting the taps. Another way to term this is P1 as the naive state and P2 as the learned state. And here, these arrows represent the transition probabilities between P1 and P2. So we wanted to simulate if this model was actually sufficient to produce something like the graded response I actually saw in the experimental data. So what this represents is two different parameters. For P1, the probability of response is 80%, P2, the probability of response is 41%. And when we model this for each individual cell, we found that for different parameters, so this would be a different cell here, we can actually recapitulate this gradual decline. So even though there's a discrete switch for each single cell, when we model it with a collection of cells, there's a gradual decline in the contraction probability. And again, these are the same axes. On the x-axis, we have the stimulus number, and on the y-axis, we have the fraction of cells contracting in the population. And once more, we basically overlaid the simulation onto my actual experimental data, and we found that it's a pretty close match. So we can be pretty confident that this two-state model can account for the habituation curve that I see in my experiments with the actual cells. And then basically from the data I got, we calculated what are the probabilities of contraction in each state for a high force versus a low force. So as I mentioned with the habituation device, which is Arduino controlled, we can change the force and the frequency level administered to the cells in the dish. So with a high force, there's actually a higher probability of contraction even in the learned state compared to the low force. And in just a little bit later in the talk, I'll talk about other differences we see in different forces applied to these cells. So the next question leading into it is, which factors influence switching from one state to another? Force is one that I just mentioned, and I'll mention a few others. To first answer this question, I was interested in forgetting. So you saw in the two-state diagram, there are two arrows. There's a forward transition from P1 to P2, but there's also a reverse transition from P2 to P1. And the reverse transition, another way of describing it, is forgetting. So to measure this, this is the experimental schema over here. We stimulated the cells until they were habituated. So these little spikes represent taps that the cells are receiving. And then we pause the stimulation for 15 minutes, and then we resume the simulation afterwards. So the initial response, and this is on the y-axis, there's a fraction of cells contracting. Again, this is population level data. And then on the x-axis, we have the different stages of the experiment. So the initial response, most of the cells contract, 60% of them contract. Once they've habituated, almost none of them are contracting anymore. But then after a 15-minute pause, they resume their initial contraction probability. And this tells us a couple of things. First, stentor forget within 15 minutes because they're resuming their initial contraction probability. And two, stentor remain in this habituated learned state, P2 in other words, as long as they continue to receive stimulation, as long as they keep getting the taps. We also wanted to test if stentor can anticipate stimuli, sort of like if you, one stressful story, personal story I have is when I had to set an alarm to catch a flight last week. My phone broke down that morning and my alarm actually didn't go off, but because I sort of have an internal clock, I can anticipate what time it is. And so I woke up at 3 a.m. anyway, despite my phone not going off. So can stentor do the same thing is the big question. And it turns out, no, they cannot. So the way we arrived at this answer is we repeated the stimulation to stentor cells and at 1 minute increments, and then we paused it and measured how many of them still responded at the same 1 minute increment despite not getting an actual stimulation. So now moving to the results on the y-axis, we have again the fraction of cells contracting on a population level. And we have the number of cells that contracted at the last stimulus. So most of them are still contracting. They have not habituated yet. And 1 minute after the last stimulus, they're not really contracting because they didn't actually get the stimulation. So they're not contracting in anticipation of getting another tap. So now I'll go back to some of the forces that we were talking about before. The forces that I showed in the previous experiments are what I'm going to call low force. So stentor habituate more easily to low force intensities, and that makes ecological sense because predators are more likely to be more aggressive, but something harmless like ripples in the pond are more likely to be low force. So that's something that should be easier to habituate to. So just to see what the impact of high force is, we changed the settings on the Arduino and did other experiments at a higher intensity. So on this plot here, on the x-axis, we have stimulus number, and on the y-axis, we have the population contracting, so the fraction of cells contracting in the population. And if you can compare this to the previous data I showed, the habituation still occurs, so stentor can still learn under high force conditions, but they're less habituated as a population by the end of the 30 trials. So still a little under 40% of them are contracting rather than under 20% that I showed earlier. But then I was still interested in whether under these high force conditions, the single cell analysis still holds up. So to do this, I repeated the procedures before. I analyzed each single cell in this population, and I looked at their contraction behavior at each single time point. And the black squares represent instances where the cells got a tap and contracted. The white squares indicate when the cells got a tap and then did not contract. And again, just visually, you can see that there appears to be a discrete switch from when the squares are mostly black to when they're mostly white. And we followed up this by doing the same procedure of implementing an unbiased algorithm at every single time point in this experiment and comparing before and after using a Fisher's exact test. Is there a significant difference in contraction probability? And we found that indeed, yes, there is. There is still a single switch that occurs for each cell in this population, but that switch occurs at a different time point, which accounts for this graded population level decline. And this is a similar analysis showing that there's only a single step. When we do the Fisher's exact test, there's a significant difference in contraction probability before and after the step. And then when we analyze all of the time points before the step, the pre-step probabilities, there is no significant difference, and same for post-steps. So basically repeating the analysis before and after the step that we found. And the conclusion is that there's only a single step, there's not multiple steps. So then the next question here is, how does the frequency of taps influence their learning? We already talked about force, but perhaps the frequency of taps also changes the transition dynamics. So I'm just going to show you this video of stentor constantly getting tapped. Before, the experiments I showed were at a frequency of 1 tap per minute. But here, the stentor are getting forces at a frequency of 1 every 1.2 seconds. You can see once they got the first tap, they all contracted, and they're repeatedly getting taps at about 1 per second. The stentor usually require 45 seconds to a minute to completely re-extend. So there's not enough time for the stentor to re-extend in between the taps they're getting. And you can see from the water shaking, that's each impulse is causing the water to shake. You can already see that some of the cells are not contracting. And normally it takes the cells 30 minutes to 60 minutes to fully habituate. But in this video I'm showing you, we're under a minute now, and some of the cells are already habituating. For example, this one here is re-extending, is no longer contracting in response to the force. So I'll fast forward a bit. You can see even more of the cells are remaining in the extended state. They're no longer contracting, and it's only been 2 minutes into the experiment. So they're learning really quickly once they get high frequency stimuli. And so here we're at the 3-minute mark, and the majority of the cells have learned. So it's quite remarkable that this happened in under 5 minutes when normally the cells require around an hour to learn. So this is what this looks like when we plot it. So on the x-axis, we have stimulus number, and on the y-axis, we have the fraction of cells contracting. And as expected, upon receiving the first tap, almost all of the cells contract, but by the end of the experiment, in this high frequency experiment, almost none of the cells are contracting anymore. So there are 2 ways to look at these data. So we can either have on the x-axis the stimulus number, the number of taps they're getting, or on the x-axis, we can have time. So I'll walk through both of these ways of looking at these data. So in red here, we have the high frequency data when they're getting the tap once every 1.2 seconds. And in blue here, we have the normal conditions. They're receiving a tap every 60 seconds or every 1 minute. So as the stimulus number increases, there actually appears to be faster learning when the normal group. So 30 taps delivered at a frequency of 1 per minute actually leads to more habituation than 30 taps delivered at a high frequency of 1 per 1.2 seconds. So this tells me the stentor are not counting, because if the stentor could count to 30, it wouldn't matter what the time interval in between these taps were. These curves would be the same. And in this second plot here on the x-axis, we have time. So again, in red, we have the high frequency experiment. And when we look at time on the x-axis, on the other hand, there's a much more rapid decline in the high frequency experiment versus the normal experiment. So I'm still in the process of making sense of these data, but they tell me a couple of things. One, like I said, the stentor not counting because 30 taps means different things depending on the time interval in between each tap. And secondly, when just looking at the time rather than the stimulus number, the high frequency group actually learns faster. So there might be some combination of these two factors, stimulus number and the time in between might be involved in memory consolidation. Let me see, I see there's a Q&A question. In the habituated state, is it right to think the organism is in a form of fetal position, essentially in full defense, but not able to do much else? In real life, when would that extended non-reaction time be dangerous itself? So in the habituated state, I wouldn't call it a fetal position because in the habituated state, the cell is actually extended. So when the cell has learned to ignore the stimuli, they are no longer in that ball-like contracted state. So I guess the second part of the question is the cell in the contracted state in full defense and not able to do much else. Because the re-extension time from the ball-like state is pretty quickly, I would say that the cell is still pretty functional. I don't know if the cell can still feed or take in food through its oral apparatus while it's still in that contracted state, but I would say that it's still a fully functional cell. It's not gone into a non-functional mode when it's contracted. So inspired by my own experiences cramming for exams, I found that when I really cram and try to do high frequency learning on myself, I forget all the information immediately after the exam. So my question was, do stentor forget more quickly after high frequency training compared to normal training? So see for yourself. So in this experiment, so this is the tail end of a high frequency experiment. So they're still getting the tap every 1.2 seconds. And the tap stopped now. And the timer is starting for 30 seconds. I'll fast forward a bit. And then I want you to watch for what's going to happen to the cells once they get the first tap after 30 seconds have passed. All right, so there it was. And you can see almost all the cells contracted. I'll play that contraction one more time. So there it got the pulse and then almost all the cells contracted. So while normally the cells can remember for at least 15 minutes or a few minutes after training, here the habituated stentor forgot within 30 seconds. So to summarize this part of the talk, I just wanted to wrap up a few things. First, it's really amazing to me that single cells can learn despite not having a brain. And what we looked at is finding out what are the individual cells doing to learn within a population. So there's a population graded level response that we see from the cells contracting in a dish repeatedly using this habituation device. But actually this population graded response emerges from single cells undergoing step-like switches suddenly, eureka moments for each cell that occur at a different time point that lead to this population level decrement. So we can model this as a two-state model between the responsive naive state when the cell is constantly contracting to the habituated learned state. And this transition from responsive state to habituated state and vice versa is influenced by the force and frequency of stimulation. And just to pause here, I mean, one of the things that I really love about these experiments is I feel like it's an ode to heterogeneity within a population. I always like to think about, I mean, I know that in science, we often talk about not anthropomorphizing the creatures that we work with. I personally think that by calling some of the, by ascribing some of that term anthropomorphizing to a lot of this research, it's sort of giving humans the monopoly on learning and intelligence, when actually there are multiple other creatures across the tree of life that are capable of complex computation. And so when I see these cells that are learning in step-like fashions at different time points, it makes me think about, how kids in a classroom all learn at their different pace. And so that, to me, if a single cell, if they all learn at the same pace, for example, if they all switched at the same time, you can imagine how that would be actually very dangerous for the population in a predator situation. Because if they're all equally contractile at a certain time point, some of them might not escape when they need to, or some of them might be responding excessively to harmless ripples. So I really like to see all of these cells, they respond and learn at different paces. And for me, that's one of the beautiful parts of biology, just the heterogeneity of it all, even in terms of their learning. Okay, this is a really good question. How has this work changed the way you think about how we define intelligence? Yeah, that's a really great philosophical question, and I'll talk a little bit more about that at the end. But just to begin to address it, I have a very broad view of intelligence. I'll start by my definition of learning. I define learning as an adaptive updating of system processing in response to new information. So in the case of stentor, they're getting new information about how harmful or harmless a tap is, and it's adjusting its behavior, the contractility accordingly, according to this new information. So that definition of learning is general for pretty much the whole tree of life, and even non-life forms like computers. So I would consider learning as a subset of intelligence. And I mean, intelligence, for me, it means sort of possessing knowledge about the environment. And every creature that does complex computation has some form of intelligence, because it's figuring out how to navigate the environment to survive and reproduce. So it's changed the way I came from a neuroscience background, and I spent my undergrad studying how the brain works. And I love the brain. I think it's so complex and fascinating. But one of the reasons why I transitioned away from studying neuroscience is I felt that the brain is not the fundamental unit of intelligence, and there's so many other different life forms that perform complex computations, and learning about how cells learn has just hit that point home. Okay, so now to this next question: where and how does learning occur inside the cell? And this is still very much an open question that I'm working on in the lab. So the things I'll show here are very preliminary and just things that are on my lab bench as we speak. So I first want to mention that stentor mate via conjugation. Sorry, this got cut off a little bit, and exchange cytoplasm. So stentor can reproduce sexually and asexually. So they can, the asexual method is they reproduce by binary fission, so they just split into two. But when under stressed conditions, the stentor mate via conjugation. And here you see two mating stentor, and they're exchanging cytoplasm and nuclei. And so this is what that looks like under a microscope. Two stentor that are exchanging cytoplasm. And the reason I'm interested in this is the cytoplasm must be the locus of learning for stentor. These creatures, these cells don't have a brain, and the molecules that produce the learning that I've shown must reside in the cytoplasm, which is this jelly-like substance inside the cell. So I was really interested in figuring out how mating occurs in stentor, because I thought this could be a really interesting conduit of a memory transfer in these cells as they exchange cytoplasm. So it was actually a very big question in the field, how to induce mating in stentor, because it was really hard to get this done reproducibly and in large quantities. So the way I figured this out was an accident. I remember the day because it was a very surprising finding for me. It was September 7th of 2022 that I opened my drawer and I put my glass jar or my glass bowl of stentor under the microscope, and I saw hundreds of stentor mating, and I'd never seen them at such quantities before. And I realized this was the bowl that I'd forgotten to feed for a couple of weeks, and I just sort of left it on my bench. And by accident, I tried to retrace my steps. How did all this mating happen in this one bowl that I had? And I came up with this protocol to reproducibly do this in other cultures. And it all came from my neglectful accident of forgetting to feed the cells. So basically, you make sure that the stentor are fed very sparsely. So every 7 days instead of, I would usually feed them every 5 days or so. And then mating should begin after 14 days of relatively sparse feeding. And the culture also needs to be of a high enough density so to produce the mating phenotype. So there are two factors in short that produce this mating phenotype in stentor: high density and starvation conditions. So it seems that when the stentor are under these stressful conditions, they want to maximize their chance of survival. And so they start exchanging or maximizing genetic diversity by mating. So once I was able to induce mating, I wanted to see how they respond to mechanical taps. And you might have just seen, so all of these are mating cells in the Petri dish. They're all attached to one another. And when they get a tap, they contract in sync. And I've never seen one cell contract without another cell also contracting. So this could mean one, either their contraction apparatus is linked up once they start mating, or it could mean that their learning apparatus is linked up while mating. So then I looked at whether habituation is still possible while mating is happening, and turns out mating can still occur, although to a lesser extent. So at the beginning, all the cells contract, and by on the x-axis, we have the number of taps the cell is getting, and on the y-axis, we have the fraction of cells contracting. And by the end of the experiment, the probability of contraction has gone down, but certainly not to the level of single cells habituating, because before it was around 20%, if you recall from my earlier slides. And this could be because when the cells are tied to each other, it's sort of like those potato sack races. You know, if one person falls, then they drag the other person with them. So it could be that it's just a higher contraction probability overall, because when one cell is contracting, it sets off a fluid motion that forces the other cell to contract. They're sort of tethered together. So I wanted another method to tease apart what is happening here. So I'll get to that in just a second. But I first want to mention that stentor are capable of another amazing feat other than learning, and that is regeneration. So when I'm trying to find out where in the cell, where in the cytoplasm this learning is located, I can try to localize the brain of the cell by cutting it into different pieces and measuring its learning behavior. So this is an alternative to the mating method that I showed before. So this is a really cool video done by Cindy Tang at Stanford, showing that a stentor cut in half can fully regenerate into 2 new stentor. So they were cut by a microfluidic guillotine, and we have each side of it regrowing over the course of several hours. So by around 10 hours, you have a fully regrown cell. So the stentor are remarkably resilient and can fully regenerate. And the time scale for this is around 8 to 10 hours. So the stentor are cut, and then the head grows a new tail, and the tail grows a new head. And this process happens over around 8 to 10 hours. So I wanted to do experiments on just the posterior halves of these bisected cells. And the reason I chose the posterior half and not the anterior is because the cell needs this holdfast, this tail, in order to stick to the bottom of the dish, because that makes the contraction easily visible when I'm imaging it using the USB microscope. If I were to use the anterior half, these heads would just be floating around. They wouldn't be able to contract because the contractile apparatus is in the posterior segment. So these are just, I mean, it's a little bit hard to tell at this magnification, but these are just the posterior halves. I've just cut them in half, and I'm trying to find out whether they can habituate at the same level as normal cells. So this is at time 0. They've just been put in the dish. The experiment has started, and I'm looking at their contractility. So they just got a tap and all of them contracted. So no deficits there with the posterior half. But now I'm looking at time 60. I want to see how they respond after 60 minutes of training. And they've been receiving 1 tap a minute for 60 minutes. And I'll play that impulse one more time so you can see. You can see that most of the cells contracted, but there are a few of them that remain extended. It's hard to see because they're smaller since they've just been cut, but habituation is possible in only the posterior halves of the cell, although it occurs more slowly. So this tells me that there's something, when I'm trying to localize where this learning in the cell is happening, just the posterior half is sufficient for certain cells to complete the learning process, although there might be something in the anterior half that actually facilitates the speed of learning. One dream experiment that I'm still in the process of developing the techniques for is figuring out where learning occurs by doing surgery on the cells. So one way to do surgery on the cells is to graft different cells together. And so here's a big cell grafted to a small cell, 2 different cells grafted, basically different ways of fusing cells together. And I want to see if I can take a cell that has been trained and fuse it to a cell that is naive and see if any memory transfer occurs. And that memory transfer would be the output I'm measuring is the contractility of the newly Frankensteined cell. So this would be because the limitations of mating are that I have to induce an entire population of cells to mate. And also when the cells are mating, it's hard to tell whether their learning apparatus is linked or their contractile apparatus is linked. The memory transfer might be the most elegant and effective way to actually figure out which part of the cytoplasm of a cell is important for learning. For example, if I only fuse the posterior half to a naive cell, would that be sufficient for a memory transfer? So I'm really looking forward to doing this experiment because I just think it's science fiction happening in present day. I was also interested in finding out how other types of stimuli impact stentor behavior. I've only talked about mechanical stimuli so far, but stentor live in a messy pond environment and they're interacting with all sorts of complex challenges. And one of those challenges is light. So stentor have negative phototaxis, which means that they swim away from light. And this has consequences for stentor learning. So other people have done experiments to see if stentor can habituate in response to light, and turns out they can, but it's a separate pathway than habituating to mechanical stimuli. I was interested in how stentor's exposure to light affects their clumping behavior. And what you see in my Zoom background is actually the stentor have aggregated in a rosette formation in a clump. And I wanted to see if that is somehow related to the presence of light. And so basically the experiment was leave a dish with stentor in it exposed to light and compare it to a cell or a dish of cells completely in dark conditions. And it turns out that the cells in the light conditions are much more likely to show these rosette formations because it appears that the stentor are congregating in the darkest part of the Petri dish, and they're basically all trying to get away from the light as much as possible. So it induces some kind of social formation. And this is really interesting because there's a recent preprint that came out about how stentor position themselves in these congregations, in these rosettes, to actually help their feeding behavior. So in some of the earlier videos, I showed that stentor have this row of cilia, these hair-like appendages on their oral apparatus, the top of the trumpet. And this helps them take in food from their environment. And when the stentor are in a social group, they actually, the slow feeders position themselves near the fast feeders to take advantage of the fluid flow from the fast feeders and basically hijack it so that they can get more food of their own. And then they switch places constantly during this aggregation so that they can maximize their food flow. So I just think it's still a work in progress, figuring out all the different ways in which light affects stentor behavior. But I'm really interested in the possibility of social behavior and teamwork in these cells when they're moving away from light. So now that I figured out how light can impact my learning experiments, I created this very low-tech setup to minimize light and keep the stentor nicely spaced for my learning experiments, basically using old lab coats in the lab to create a curtain around the habituation device. So I found that low light and low temperature conditions promote stentor anchoring. So they actually stay in this well-spaced condition because the data are hard to analyze when they're all clumped together. So you can see here, this is an example of a normal ambient light conditions. There's still some cells swimming around and they're all sort of positioned next to the edge of the dish. And here's low light conditions. Most of them are anchored and they're nicely spaced. So that's why it's conducive for my experiments. I don't want a potential confounder for some of these experiments to be the cells are just overlapping and one of the cells' contraction is setting off a fluid wave that makes the neighboring cells contract. So all of this is great, but I've been thinking about this last question lately. Why should we care about pond scum? I was giving a presentation a couple of weeks ago, and one of the members of the UC Regents came up to me afterwards and said, "Well, I'm glad someone's interested in pond scum." And I was thinking, well, indeed I am, and I think it's something that a lot of people should care about for these following reasons. So, although stentor might seem like an esoteric, mythical creature of Golden Gate Park, they can actually help us understand human disorders of habituation. So there are actually several conditions that a lot of humans experience that are related to habituation. ADHD, autism spectrum disorder, and obsessive compulsive disorder are all related to habituation. For example, autism spectrum disorder, the little experiment I had you do on yourself with noticing the feeling of your clothes on your skin, that's something that most of you probably habituated to over the course of the day. But someone with autism spectrum disorder might be less inclined to habituate to the feeling of clothes, so would feel constant discomfort from the irritation of clothes rubbing against the skin. Another example in ADHD is hyper distractibility, because some of the ambient signals, tapping in the background, the sound of a whirring fan might be something that a lot of people habituate to, but maybe someone with ADHD would be less inclined to tune that stimuli out. So habituation would be impaired. And obsessive compulsive disorder, the compulsions in OCD arise from a lack of habituation to anxiety. So it's an anxiety disorder in which there's a lack of habituation to that feeling of pain and discomfort. So by studying habituation at the level of a single cell, we can get a better understanding of these human disorders of habituation, because a lot of psychiatry is done at the level of circuits of neurons in the brain. But we don't know a lot about how individual cells within those circuits are actually doing computations that contribute to this overall dysfunctional network. So by studying learning and computation at the level of a single cell, we get a more granular understanding of how these circuits might work in the human brain. It can also help us learn how to train other cell types. So here we have a bunch of white blood cells that are going towards an attractant. So white blood cells are involved in a lot of our immune response. So what we've done with stentor is basically shown that we can train cells, and we have a reproducible way of manipulating their behavior. So once we've learned how to train one cell type, we can see if we can actually learn how to train other different cell types like these white blood cells to train them to do different things inside our body, if we can understand general principles of cell behavior and how they are performing these computations. They can also help us learn about how to train cancer cells. So here we have breast cancer tumor cells that are progressing. They're migrating to get in a sheet. So cancer, I mean, is obviously is terrible, and we're still trying to figure out how to curb the progression of some of these tumors. And if we can train cells to do our bidding and go in a different direction, or train them to habituate to different stimuli, we can perhaps change the behavior of their migrations within the body. So although we're starting with pond scum and stentor, if we learn the principles of habituation and learning within these single cells, hopefully we can translate that to different cell types like tumor cells and white blood cells. And I also, I am working towards my PhD, and a PhD stands for a doctorate in philosophy, but I feel like the philosophy part is often neglected in the sciences, but it's something I'm very interested in. One of the things that fascinate me about stentor is that these cells defy dualism. Dualism was an idea put forth by a lot of people, but most famously by Descartes, suggesting that the mind is separate from the body. The mind is something with a more ethereal spirit that is different from a tangible physical form. However, stentor flies straight in the face of that knowledge, because in stentor, the mind is the body. There is no separation because the cytoplasm is the locus of learning. There's no separation because they don't even have a brain. So I think stentor is a great system for armchair philosophers to finally take a seat at the lab bench. And finally, this is my main motivation for studying these questions. I'm really interested in learning about the origins of intelligence. So this picture right here is actually my desk at home where I'm sitting right now giving this presentation. I'm sitting, my laptop is at this desk as we speak, and above me is this giant poster of the Tree of Life. And this poster inspires me every day because I'm really interested at the roots of it. Up here we have humans, but what paltry creatures we are in the face of this wonderful menagerie of other fantastic creatures. So I'm interested in the base of this Tree of Life and how complex computation could arise even in these life forms here. And unfortunately, this picture doesn't have stentor on it, although they should draw it in because it's very charismatic. But I'm interested in how, if we can understand how even a single cell can learn without a brain, we can understand how complex computation made its way through evolution up these other branches of the Tree of Life. Because life began with computation. And so I feel like the origins of life and the origins of intelligence are the same question. So a brief touching on current and future directions. I didn't talk about this today, but hot off the press, I'm finding out new molecules that are involved in habituation and stentor. So I talked about short-term habituation under the scale of around 60 minutes, but I've also done experiments with training the stentor for several days at a time. In fact, whenever I go on vacation, I like to keep the cells on the device getting tapped repeatedly while I'm out on Christmas break. And so that's several days, 2 weeks at a stretch, and I come back and I see how smart they are. And so by using various drug treatments, I've found some of the molecules that are involved in habituation, and I'm now following up with proteomics to see how those molecules are conserved across different species and whether they're similar to the molecules that we use for short-term memory. And also, speaking of different species, I recently had a haul of stentor from Golden Gate Park, and I found other species besides Stentor ceruleus that I focused on today. So as I showed you in the Tree of Life, there are other single cells out there that are similar to stentor, and I want to see if they can learn in the same ways, if there's some conserved mechanisms of learning that are common to all of those species. I also want to see how light interacts with the mechanical stimuli, if they actually work together in promoting learning, or if there's some sort of competition between those pathways. Social learning, if stentor are able to communicate with each other in some ways. Associative learning is a different form of learning. Basically, the Pavlov's dog experiment. In Pavlov's dog experiment, basically the dog was presented with food and a ringing bell at the same time. And the dog eventually over time learned to associate the ringing bell with the food and began salivating with just the ringing bell without the food. And this is thought to be a complex form of learning limited to creatures that have a brain or at least a nervous system. But I want to see if it applies to stentor as well. And finally, one program I'm really excited about is called Stentor in Every School. And I think in some of the courses I've taught, lots of elementary school children have come in, and it's been amazing seeing their reaction to seeing stentor under a microscope for the first time. And it's a really great way to show people that cells are more than just a bag of chemicals. They're really complex, intelligent creatures. And so we started with some of the other students at San Francisco State University, a program called Stentor in Every School, starting to bring stentor into elementary school classrooms and having the kids actually do research. So that launched in February, and I'm excited to see what amazing these discoveries these 4th and 5th graders find. So with that, I'm really grateful to be able to show stentor to you today. And I just like to add that the next time you put on your clothes in the morning and learn to ignore them over the course of the day, I hope you give a nod to stentor for showing us you don't even need a brain to be smart. Thank you very much.